Hey, why did you eat me? Because I was hungry. If I was hungry, I would not have ate you. So why did you eat me? You can't eat me if you wanted to or not. So now you think you're better than me? I am. I'm the best. I can eat you and all your friends. The only way we can know who's better is if we know what each of us are made of. I think we should explain how grade 10s would learn. That's probably the best way to learn about each other. Great idea. I think they learn from the McGraw-Hill textbook. So we should learn from the grade 10 science textbook? Yeah. Okay, I will start. Microscopes give us the ability to see things we can't see with our eyes. Microscopes can also help people find treatment for diseases. There are many different types of microscopes. Let's start off with the light microscope. It is often called a bright field microscope because the image is seen against a bright background. It is used in laboratories to study cells. You will place a thin disc under the stage which is between the light source and the objective lenses. We would not be able to treat many diseases without microscopes. One of the very first organelles scientists could see was a nucleus. With all the new advanced microscopes today, we now know that the nucleus is made of parts too. It has a nuclear membrane, which acts as a wall to protect the inside of the nucleus. We have the nuclear pores to allow materials to pass through. And there's also the nucleolus, which makes ribosomes. Ribosomes help make proteins. The organs of our body are very similar to the organelles in a cell. Everything plays an important role. The normal animal cell contains the nucleus, endoplasmic reticulum, cytoplasm, vacuole, Golgi body, cell membrane, vesicle, cytoskeleton, mitochondria, and ribosomes. They each have their own rules, which are very important. The cell membrane acts like a wall with a door. It separates the inside of the cell from outside of the cell. It also allows certain materials to flow through, into, and out of the cell. It separates the inside of the cell from the outside. The cytoplasm contains all of the organelles. It also contains sugar and water. The cell's activities are fueled when the mitochondria releases glucose. Proteins are made with the help of the ribosome. These proteins are needed for the cell's activities. These ribosomes can be attached to the endoplasmic reticulum or float around in the cytoplasm. The endoplasmic reticulum packages proteins and sends them around the cell. The Golgi body's job is to organize proteins to send them outside of the cell. The nucleus controls all the cell's activities. The vacuole is responsible for storing small molecules that can be later used for transport. The cytoskeleton provides shape and movement of the cell. The cell wall is a strong structure outside of the cell membrane. Chloroplasts are also very important to photosynthesis. They trap sunlight to make glucose. The glucose is then given to the mitochondria to fuel the activities. Keep in mind that chloroplasts and cell walls are only in plant cells. The mitochondria in plant cells gets glucose from a process called photosynthesis. Photosynthesis is when chloroplasts trap sunlight. This energy is converted into glucose and given to the mitochondria. The mitochondria fuels the cell's activities. The process when the mitochondria fuels the cell's activities is called cellular respiration. This requires oxygen, glucose, carbon dioxide, and water. They learn about genetics next. In Canada, babies are tested for a genetic condition called PKU. If this is not corrected, PKU can cause brain damage. If we want to understand this, we need to know a lot more about cells. The nucleus controls the cell. It will say what cell will become what cell and how it will function. Chromosomes carry these instructions. Every species has its own specific number of chromosomes in the nucleus of every cell. Humans have 46 chromosomes in each nucleus. These are made of a material called DNA. Each chromosome contains one molecule of DNA, which is divided into segments called genes. Many genes give us instructions for making protein. DNA controls many of your features, including your hair color. DNA screening is when you test it for genetic disorders. Some disorders can be observed by a number of chromosomes. Much of the treatments for diseases is carried out by drug companies. This research can be very expensive and can consume a lot of time. Once a drug is developed, it is tried on humans. In Canada, this needs to be approved by Health Canada. The risks of losing money are really high for the company since the drug may not work well outside of the lab. Cloning is another solution to creating new identical organisms. This has been used by gardeners for centuries. Gardeners take cuttings from a plant and rooted them, which created identical copies from the previous. This can also be done with mammals. Now we should talk about cell division. Cell division is when two parent cells divide themselves into daughter cells. Cell division is a main process for a single-celled organism such as bacteria. For a multicellular organism, Now we should talk about cell division. Cell division is when two parent cells divide themselves into two daughter cells. Cell division is a main process for single-celled organisms such as bacteria. For multicellular organisms, cell division is when a fertilized cell becomes... For multicellular organisms, cell division is when a fertilized egg becomes an adult with millions of cells within the adult cell. Replacing lost or damaged cells in multicellular organisms is also a process of cell division. An example of this would be when you cut yourself, the skin would come back, and after a short period of time, it will look as if nothing happened. Another very important reason why cells would divide would be if they are too large to do a task efficiently. The cell membrane plays a very important role in this. When you eat food, it gets broken down by the digestive system, then sent to every single cell. When they arrive to the cell, they must enter through a cell membrane. These food molecules pass through the membrane with the help of diffusion. Diffusion is when the molecules of a higher area concentration move to an area with low concentrations. An example of this would be when you peel open an orange on one side of the room. The smell would eventually go to the other side as well. This is because on one side there is a higher level of concentration from the other side. This is an important process used by the cell membrane because when the cell needs materials such as oxygen, the oxygen will diffuse across the membrane from outside of the cell. Cells are most often surrounded with molecules like water. Water moves from an area of high concentration to an area of low concentration. This process is called osmosis. There are two stages in cell division: mitosis and cytokinesis. Let's talk about mitosis. There are many phases in mitosis. The first one is prophase. During this stage, the chromosomes move in different ways until they are finally thick enough to be visible. The nuclear membrane starts to break down. Also, two organelles called centrosomes go in opposite directions. The next phase is metaphase. In this phase, the centrosomes are in opposite sides of the cell, while the chromosomes move to the center. The next phase is anaphase. During this stage, the two chromatids break apart. The spindle fibers react from being stretched, and after the chromatids separate, they each become into a chromosome. At the end of this stage, the cell has double the chromosomes. Telophase is the final stage. At this stage, the spindle fibers disappear. The cell membrane forms around the two new daughter nuclei. The nucleolus reappears and the cell is ready to divide. Cytokinesis completes the process. Once this process is complete, there should be two new cells. This is just a bit different in plant cells than animal cells because plant cells need a new cell wall. In the plant cell, Golgi body creates small vesicles which carry out materials needed to create a new cell wall. They line up between the two new nuclei, creating a cell plate. The cell plate grows outward and joins the old cell wall, dividing the cytoplasm into two. The new cell membrane forms inside the cell walls. This is done millions of times to grow from a single cell to a multicellular fetus, and then finally into a human. The cell cycle is also very important. The first part of the cell cycle is cell division. The second part is interphase. Cell division is when the nucleus divides into two and then divides into two daughter cells during cytokinesis. During interphase, cells do what they're supposed to do, such as producing proteins, taking in oxygen and glucose, cellular respiration, and removing wastes. Cell cycle checkpoints are used to control the timing and rate of cell division. These are very important to normal growth. Too many or not enough cells in one body part can lead to serious problems. There are three main points at which cell checks its growth. These stops will not let the cycle continue if there are not enough nutrients to support the cell growth, if the DNA has not replicated, or if the DNA is damaged. The next topic is cancer. In the cell cycle, cells start off normally, but a few are transformed to ignore the checkpoints. Most cells only divide when they are attached to a surface. If they are not attached, they stop dividing. Cancer cells have a mutation that allows them to continue to divide. They don't stop at the cell checkpoints, but instead divide themselves repeatedly. This forms a clump of cells called a tumor. These cells can later become cancer. Some cancers spread to other parts of the body. In a healthy tissue, cell division is controlled so the rate of cell division is equal to the rate of cell death. Sometimes cells start to lose their rate of cell division and begin to divide more often than usual. All the cells that have been divided by the cells that have abnormal rates of division also have divided at a rate of speed that is not normal. This abnormal division increases the size of the tumor. If these cells change further, they can produce cancer. Cancer cells invade other cells. Some of those cancer cells move to the circulatory system and can spread to other parts of the body. I think it's time to learn about cell specialization. This is a process in which cells develop into cells with specific functions. Plants are always making specialized cells. Groups of specialized cells form into tissues. Groups of tissues work together in organs. Special cells called meristem cells are responsible for this growth. Meristem cells produce new cells constantly. These cells form into three types of tissues found in the body of the plant. They are the dermal tissue, ground tissue, and the vascular tissue. The dermal tissue is outside of the plant's organs. It protects the inner tissues from damage and exchanges water and gas from between the plant and its environment.